We're here at the CROI, 21st CROI, here in cold Boston, talking with Lynne Mofenson, who is from the NIH. And she is going to talk about pediatric issues. She's in charge of the branch. I'll let you describe your title there at the NIH.
Sure. So I'm branch chief of the Maternal and Pediatric Infectious Disease branch at the National Institute of Child Health and Human Development. A big mouthful of words there.
Almost sounds like a German word. But it's so important that you discover that we cover this with you today, because we're at a landmark point in research in pediatrics, and I think let's go back just a little before that and then tell us about that.
So it's really been a paradigm change, you know, since I first started doing HIV work in the early 1980s. When I came to NIH in 1985, 1 out of every 4 HIV-infected mothers transmitted to their child, and about 50% of HIV-infected kids died by the time they were 2 years of age. And the very interesting thing is that in February of this year, it's the 20th anniversary of the results of PACTG 076, the trial that gave AZT to pregnant women during pregnancy, during labor, to the newborn, showed 70% reduction in transmission, something that no one thought we would really see, and resulted in dramatic changes in terms of pediatric infection in the U.S. Within, I would say, 2 to 3 years of the results of that study, transmission in the U.S. went from 25% down to 3 to 5%. Quite amazing.
So and you had a large part to do in that work.
Well, I helped with the study. I mean,
but you've been an advocate over the years for obviously in your position, but I mean you've done a lot of background work in trying to make sure that pediatrics went in the right direction.
Yes, getting attention from mothers and children is always difficult.
Can I ask you a question? Because I work as an advocate for PLWAs, and when we present or we work with pharma, the thing I'm mostly concerned about is that we actually have representation or some form where we can talk about pediatrics with the pharma. I know that in the past it's been always phase 4 research. It's they want to prove it is healthy in individuals first, then in infected individuals, and then after the drug is released or delivered to the public, it is then they do the trials in the pediatrics. Now that's being moved along more quickly.
Absolutely. Yeah. I think AZT was a good example of how not to do it. You know, it was approved for adults in 1987. It was approved for kids in like 1991. Yeah. But not just my own advocacy, but people like Elizabeth Glaser to make sure that there were funds for pediatrics. We've been able to get drugs approved for children and tested around the same time as in adults. And while there still are some delays for some drugs, there are some drugs like efavirenz was approved simultaneously in adults and in children. And we no longer require huge phase 3 trials to prove efficacy in kids. If we show that something's effective in adults, then we move back to phase 1, 2 studies showing safety and appropriate dosing in kids, and then it can be approved. So I think there's really been a big change since the 80s and early 90s.
And it's based on milligrams per kilogram.
Well, it depends. Sometimes milligrams per meter square. It depends on the drug. Yeah.
Well, that's good to know. I mean, I didn't even think about that. But is just as an aside, isn't that important even for adults? Do you feel? Because there's some really small people that are don't have a high metabolism as opposed compared to a 300-pound athlete, you know.
Yeah, well, I do think that pharmacokinetics of drugs are affected both by body mass index. You know, people who are more overweight may have lower drug levels. People who are small, like Thai women, may have different drug levels, and those are also important issues to be able to study. And in adult studies, this is one of the reasons why it's so critical to have women included in these phase 3 studies and included in the phase 1 studies to be able to determine if the dose is the same as in men and whether there needs to be modification for example, in pregnancy. Many of the protease inhibitors have lower drug levels in the 3rd trimester of pregnancy than they do postpartum. And for some of those, we recommend an increase in dose during the 3rd trimester. So it's really critical to do these studies, not just in children, but in pregnant women, and to also do them in breastfeeding women.
As we're doing the trials in so many ways that are designed, I mean, it makes sense for a drug company to come out with a one-size-fits-all. But apart from that, we really do have to think about how do we deliver the drug to a person that does not require the full dose. And then, of course, you have the sprinkles and you have liquid formulary and so forth. I just hate to see somebody that's just because they're a small person have to resort to pediatric formulation.
Well, hopefully we've been working with the pharmaceutical companies to develop scorable pills so that you have an adult tablet or an adult pill that you can break in half or break in a third and enable it to be given to kids, because actually in countries like Africa, liquid formulations are not so good because many of them need to be refrigerated.
Right. Shelf life and so forth.
And they're big to carry, and so ideally you would have tablets that you can use for adults that you can split for an appropriate dose for kids, and that can be liquefied in food or breast milk. And I think some companies are working towards that.
Now, just another question is, like I said, I'm an advocate for, I often bring up a lot of the issues with the pharma about, pediatric formulation and so forth. Is it like Elizabeth Glaser Foundation that is like the formal advocacy for children, or are there groups of people who are mothers and fathers of children who are considered like a CAB contingent to?
Well, we do have CABs as part of all of our clinical trials group. We have very active community advisory boards, and we also on our guidelines committees. So I'm the executive secretary for the pediatric guidelines and the perinatal guidelines, and we have community representatives that sit on those guidelines panels. And sometimes we really need to have the insight of the parent to be able to bring us back to earth and say, I can't do that, don't recommend that. Right. So they're very important members of our panels.
Well, I'm glad that we covered that, because that's really important for me to feel comfortable about that I don't have to be so much of an advocate for the pediatrics. It's already happening. I did have a fellow by the name of John Pottage, who runs the science for ViiV, has told me, I was over with the Elizabeth Glaser Foundation press conference, and him and I guess it was ViiV and a few others there in Malaysia for the last IS meeting, and he said, we don't bring a drug along that won't be formulable for pediatrics.
Good to hear that.
And I was pleasantly shocked to hear that.
Yeah.
And it is important, I think, for people to know that that is the case, because it is a really important piece of the science.
Yeah.
So is there anything else you want to bring up? Because I really feel like this is an opportunity that we don't get very often to visit with a person like yourself. And we did invite, by the way, we invited the Elizabeth Glaser Foundation, but I think they had a last-minute entry into the CROI. I'm not sure how that all worked out, but I can't imagine anybody having to make a decision a week before the CROI to either go or not.
Yeah, well, I guess the two things I would like to mention are, as I said, in 1985 we had 1 in every 4 infected women giving birth to an infected child, and today that's down to under 1%. So we've had this dramatic change, and the other dramatic change since I first started working in HIV is pediatric mortality. So as I said, 20 to 50% died by the age of 2 to 3 years when I first started working in pediatric infection. And death was like an everyday thing that you saw. And today mortality is almost unheard of with today's treatments. And we've moved from having to give, 3 drugs to even in some older kids being able to give them 1 pill. Just a dramatic difference in terms of lifespan. And I think Elizabeth Glaser's son is the best example of that. So Jake was infected he was infected in utero, actually, and his mother unfortunately died. He's in his 30s, and he is healthy, and he's out there advocating. And there's another young man who's on one of our committees, Ben Banks. He just had a baby, He and his wife just had a baby, and he was infected as an infant. So we've really seen such a paradigm change in pediatric HIV.
And many of these people will be around, hopefully, for a cure.
I hope so.
And a lot of what we learned from Mississippi baby patient is going to be applicable, or at least informative, to cure research. It's all a piece of the puzzle that will get us a little further down the road to that elusive cure that we now see a little more realistically.
Yeah, well, I hope so, but I think it's really important for people not to misunderstand what that represented. So what we had there was an infant who was likely infected in utero, but very close to delivery. And then they started drugs within a day. So it's like catching someone who's newly infected within hours.
And it could be as we're now learning with Steve Deek's patient, it could be even an adult.
Yes, but how many people do you catch within hours? And many babies can be infected earlier in utero. And if you're infected, say, in the first trimester, it may not be the same thing when you're starting drugs immediately postpartum. And so I think there are a lot of questions that need to be answered. And one thing I think that has concerned a lot of us involved in this research is that parents will say, well, look at this, my child's undetectable, we're going to stop treatment.
Right.
And
this
that
is
would be
absolutely wrong.
Worst possible thing to do. We need to prove that this actually works before people do things like that.
That would be very dangerous.
Yes.
Yeah. Well, thank you so much, Lynn. It's been a pleasure to have you here, and look forward to another. Maybe the next time we visit, maybe the next time we visit, there'll be a lot more towards a cure, and that'd be a wonderful thing.
Okay.
Thank
Good.
you.
Thanks.